Going back to the expression vector, you can see that another important feature exists, the polyhistidine tag. This is an amino acid motif added at the N or C terminus of the protein. Histidines are known to coordinate metal ions as they have high affinity for imidazole, the functional group of the his tag. Alongside this is the TEV cleavage site that is required for later on cleaving the his tag using the protease TEV. In the first video, I showed you how to express recombinant proteins in bacteria. At this stage, we obtained a bacterial cell pellet containing a recombinant protein, amongst many other proteins originating from the bacterial host. The very first step is therefore to lyse the bacterial cells to recover the proteins that are inside. For this purpose, we use sonication, a technique which consists in subjecting our bacterial cells to ultrasounds that will break the cell membrane, allowing for the release of what is inside. First, we resuspend our cell pellet in a buffer that will ensure the stability of the recovered protein and allow for its subsequent purification through affinity chromatography. Resuspension is done by vortexing the cell pellets in the buffer until the solution becomes homogeneous. As proteins may be unstable at room temperature, all subsequent steps of the purification are performed at 4 degrees Celsius or on ice. Once the pellet is resuspended, we lyse the cells with a sonicator. The probe that we plunge in our sample will send ultrasounds that break the bacterial membrane. As this process generates heat that may denature the proteins, we keep the sample on ice and allow it to cool down again for 5 to 10 minutes between each round of sonication. Once cells are efficiently broken, the soluble proteins contained inside the cells are separated from the cell debris by centrifugation, and we recover our recombinant protein in the supernatant, together with the protein contaminants from E. coli. We rely on the high amount of histidine amino acids in our protein of interest for its selection. So a very common method for selecting our protein of interest, tagged with a polyhistidine motif, is the use of immobilized metal ion affinity chromatography. Recombinant proteins expressed in bacteria are generally designed to encompass a tag that allows for the separation from the E. coli proteins using affinity chromatography. The most common used tag is the polyhistidine tag, which consists of 6 consecutive histidine residues added to the sequence of our protein, either N or C terminally. This tag binds nickel ions with high affinity and therefore allows to selectively purify our protein through affinity chromatography on a nickel column. Our protein will be retained on the column, while most bacterial proteins won't be retained. This first step of affinity chromatography is performed with the help of a peristaltic pump that allows to load our protein onto the column and then perform subsequent washes with different buffers. First, we load the entire sample onto the column and collect the liquid that goes through in a bottle. At this stage, our protein remains bound to the column while most contaminants fall into the collection bottle. Next, we wash the column with a buffer containing high salt to wash out any remaining impurities. Finally, we elute our recombinant protein by washing the column with a buffer containing a high concentration of imidazole. Imidazole is a compound that has high affinity for nickel ions. By competition, it therefore takes the place of our protein on the column and thus allows for our protein to be eluted. The success of the purification is then evaluated by STS page analysis of all the samples and fractions collected during the procedure. As seen here with a recombinant protein of 13 kilodaltons, the protein is present in the supernatant following sonication and centrifugation. And after affinity chromatography on the nickel column, it is recovered in the elution fractions as a much purer sample than in the beginning. For many applications in biochemistry and structural biology, it is convenient to remove the polyhistidine tag on the recombinant protein. This tag is not present in the natural protein, and it may therefore affect its physiological function. To remove it, the original construct design usually incorporates a protease sequence after the polyhistidine tag, so that the tag can then be removed by proteolytic cleavage. In our case, we use a sequence corresponding to the cleavage motif of the tobacco H virus protease, TEV in short. In practice, to remove the his tag from our purified protein, we incubate the protein overnight with TEV protease. As the protease is less active in the presence of divalent cations, we perform the cleavage simultaneously with a dialysis in a buffer containing EDTA in order to remove any cations that may be present in the buffer. The cleavage is done overnight at 4 degrees Celsius to ensure that all protein sample is cleaved. On the next day, we recovered the cleaved sample and pass it through the nickel column again. This time, since our recombinant protein does not contain a polyhistidine tag anymore, it won't be retained on the column and will fall off in the first collection tube. On the other hand, the his tag, the TEV protease, which contains a polyhistidine tag, and the contaminants from E. coli that bound to the nickel column in the first step will still bind to the column in the second step. So this allows for a very efficient separation of our protein from these contaminants. Again, we check the efficiency of the purification by SDS-pH analysis of all the fractions that were collected. As you can see on the gel, we notice a 3 kilodalton difference in size for our protein before and after TEV cleavage, which corresponds to the removal of the his tag, and the protein now comes in the run-through fraction as expected with a high degree of purity. And we can now perform the last polishing step of purification, which corresponds to size exclusion chromatography. At this stage, the protein is already very pure, but it may exist as different oligomeric forms. For structural studies, it's very important to work with a homogeneous sample, meaning a sample that contains only one type of oligomer. The last purification step thus aims at separating possible distinct forms of the protein. Size exclusion chromatography, or SEC in short, separates proteins according to their size. SEC columns are usually made up of a dextrand agarose matrix, forming porous bits. The protein is passed through these porous bits, and depending on its size, it will take more or less time to go through the column. Large molecules will only go through the large pores, and therefore will pass through the column faster, thus eluting earlier. On the other hand, smaller proteins will go through all small pores and therefore will elute later from the column. Different bit sizes exist commercially that provide a different range of molecular weight separation, allowing to select the column that provides the best resolution of separation for our protein of a given molecular weight. In our case, for a protein of 10 kilodaltons, we use SuperDex 75 increase column. At this stage, we first need to concentrate the protein to have a lower volume of protein and thus a less diluted, purified protein. And also to avoid losing too much samples due to too many runs on the column. Concentration is performed with the help of centricons. These are tubes that contain an upper and lower part separated by a porous membrane that only allows the passage of small molecules, but no proteins. Centrifugation will allow the buffer to pass through the membrane, while the protein will remain in the upper chamber. Thus, the protein will be progressively concentrated as the buffer is removed from the sample. The last step of purification is performed on an FPLC system that allows to monitor very precisely where the protein elutes by following the absorbance at 280 nanometers of the eluting sample, all throughout the purification. The sample is first injected into the loop mounted on the system with the help of a syringe and a needle. The rest of the purification is then piloted from the computer with a dedicated software that allows to inject the sample from the loop onto the column and then perform the elution of the sample in the desired buffer. the absorbance at 280 nanometers, allowing to detect proteins eluting from the column, is monitored by a cell placed at the exit of the column, and the measurement is directly read on the chromatogram displayed on the computer screen. The sample is then sent to a fraction collector that collects the liquid coming out of the column in different tubes. As you can see here, our protein of interest elutes at around 12 to 13 ml of elution in fractions 21 and 22. We can then recover these fractions and analyze them by SDS page to confirm that they contain our protein of interest. We can see two different peaks on the chromatograms that correspond to two distinct oligomeric forms of the protein. The fractions corresponding to these two different forms are pulled separately, and we can then freeze our protein at -80 degrees Celsius for long-term storage.